Hey guys, welcome to Module 8. Today I'm going to talk a little bit about the DNA polymerases and try to kind of put them in maybe a little bit more logical order than the book did. I'm mainly focusing on two different polymerases, Pol3, which is going to be our main replication one, which I'm going to talk about at the end, and Pol1, which is used as either a DNA repair or primer removal. So the way that all DNA is synthesized is that you actually start with a small strand of RNA, so about 10 nucleotides that are laid down, and then from there, DNA polymerase 3 is able to bind to this small primer strip to extend and generate a large amount of DNA, and then later on, Pol3 comes through to remove this. Your book kind of illustrated this, but I'm going to add in the last section. So you start with this DNA template here. You'd have your primase come in, add this RNA primer sequence. Your DNA Pol3 now comes in, synthesizes your new strand of DNA, and then your Pol1 comes in, removes this, and replaces this with DNA. So I'm going to start off talking about Pol1 to kind of get it out of the way. It's a little bit simpler of a process. So this is the structure of the actual protein. It kind of looks like a hand where you have these fingers, palm, and thumb here. And what these do is they essentially hold the DNA. The DNA is going to pass through the middle of this. The thumb will be on top and the fingers will be behind. That way it's holding it, kind of like cradling it. And it essentially runs along the DNA like this until it finds either a mismatch or it finds that primer region. When it gets to one of those sites, it's going to cleave it. So this blue region here would either be a mismatch or a small primer sequence. Your Pol1 can then cleave this, and then from here synthesize the remaining DNA that was lost. So all polymerases, in order to work, require two things, you could say 5 things, but essentially you need your dinucleoside triphosphates, so that'd be your deoxy-ATP, deoxy-GTP, deoxy-TTP, deoxy-CTP, as well as magnesium, which just allows it to bind. You need a template in order to do your DNA synthesis off of. You need a primer sequence. And then, as it says here, that you need, a lot of these have ways of removing mismatches. So that's something that is involved in DNA synthesis. So what's actually happening is your 3' OH is going to attack this. They drew deoxy-ATP. You're going to lose a pyrophosphate and extend your sequence by 1. And there's no drawing of it on this page, so this OH here comes in, does your attack. It's basically NSN2 type reaction. You're going to lose this pyrophosphate, and now you've extended and you have a new OH, which can then come in, do another attack, and allow you to extend your chain that way. Okay, so this is more focused on, I would say, Pol3. So you're going to have a replication fork. So this is when you're doing large amounts of DNA synthesis. And this is the kind of stuff that you guys probably learned a long time ago, that you're going to have a leading and a lagging strand, one of which will be all going in one direction, the other one making these short little snippets, which are called Okazaki fragments. I think there's a drawing of it here. Yeah, okay. So these Okazaki fragments, which then another DNA polymerase comes in and stitches them all together. But essentially you have this fork region here you're going in one direction and the other direction at the same time. And I'm going to talk a little bit about how that exactly happens, because I think it's a little bit confusing. So, yeah, this is just saying the different things you have, which I believe are on this page. Yeah. Okay. So, our Pol 3 consists of 3 different things. So you have a DNA helicase, which unzips your DNA. You're going to have these clamp loaders, which are basically there to just align your DNA with your polymerase, make sure that things are running smoothly. And then you're going to have these polymerase cores, and these are where the actual synthesis happens. And you're going to have 2 different ones. You'll have one that's dealing with the leading strand and one that's dealing with the lagging strand. So the one for the leading strand is pretty straightforward. And what's happening is that as you're moving, as you're unzipping, you are slowly synthesizing, not the greatest arrows, this blue region here. And I think that's pretty straightforward. As it goes along the line, you're able to synthesize more. For the lagging strand, I think this image is a little bit confusing because this isn't exactly what happens. The way it works is you have what's shown on the next slide. So you have this kind of, they call it a trombone slide, because if you were to do what was on the other page, this part of the protein would be just like constantly moving away, and it would get so far away from the DNA polymerase that you wouldn't ever be able to like reconnect. So that doesn't really make any sense. What actually happens is that you get these kind of, they call it this trombone slide, where instead of the polymerase portion here moving, your DNA is moving this way, kind of like pulling a string, and you're able to get all the way from where it started to the connection of the next part over here. So it's kind of like doing that. They call it this trombone slide because it's constantly getting longer as you're making this next Okazawa fragment. Okay, I hope that was helpful for you guys. If you guys have any other questions, please send me an email. It's been great working with you over this semester, and I hope you guys have a great last week. Thanks.